we discuss in this book how the Bloom filter defined in mathematical term, okay, and how to utilize, how to develop the Bloom filters, okay. So the subset in the DNA sequence, usually people call it chimer, yeah. You can ask ChatGPT what it is, but for example, I have ACTG. If I say two-mer, so I have AC, CG, all of the combination of two uh, nucleotides inside the long sequence. But for you to continue to read, make sure you have understanding what is chimers. Okay, you can explain what is chimers in genomic. Is it like a substring, or imagine the DNA sequence is a longer string and the substring with 3k equal 3 is 3-mer. So including what is inside is a 3 nucleotides. Okay. So this is the implementation. If you want to develop Bloom filter, there is many library. In this case, we are using plain implementation using Rust language. Yeah. So you can try to run the code. I hope you already prepared your Rust environment in chapter 2. And also you can combine with Nextflow in case you want to make the code like a workflow, uh, contain many process inside the workflow. So the Bloom filter implementation, usually implementing using some kind of data structure, and there is some functionality inside, like creating new, insert new data, okay, making the hash, okay. So hash is compression, so long sequence you can compress into shorter, okay. So your data structures that contain the long sequence can be lighter, smaller, okay? And it's also give you some random samples. So instead of we read from FASTA or FASTQ, we just get synthetic data from random DNA. So this code is, I hope it's working well. If not working, please report in the comment. If you compile, when you compile the code in Rust, make sure that you put the right dependency in the cargo.toml file. Okay, so let's continue. So in the good thing in Rust is that you can parallelize easily using Rayon. Yeah. So Rayon is a parallel iterator. So if you want to make the code high performance, because the sequences are very big data, so you can parallelize using Rayon Crate. Okay. You can also implement without Rayon, but there is a significant performance difference if you parallelize the code. Another important data structure, other than Bloom filter, is the Bruegel graph. Yeah. So this is also hash-based data structure like Bloom filter. So, but it's in terms of graph. So it's a graph data structure. So this is, you can read further how to develop the graphs in the following section. But in the section 3.1, I introduced the comparison between hash table, Bloom filter, and Bruegel graph from Wikipedia. Okay. And you can also see the basic implementation of min hash signature, min hasher. I hope you try, you learn more from the code, and you experience how to run the code, how to modify using AI. So in this time, if you really understand the data structure and algorithm, how it works, you can always ask AI to write the code, but you have to use your common sense if the code is good or hallucinated. Sometimes AI is still hallucinating. Okay, so make sure you have a good understanding in Rust language or Python language. So once you get the code from the AI, you can analyze the code. Okay. Other good way is to asking the AI to always give command or documentation in the code so you can easily assess, for example, the similarity, right? This function, given two hash signature, it checks is it similar or not by doing some match operation, okay, and give you the similarity. So this is good practice to always give command to the code, okay? So try the code, practice implementing a basic algorithm. In reality, usually if you have sequencing device like Illumina or Oxford Nanopore, usually they have SDK, software development kit, for you to manipulate the data from them, yeah? Or you can also use third party like 10 Time Genomics or other companies who provide computational platform to run or to read and store the data of DNA into data structure. Okay, but the common use data structure for that is Bloom filter and also the Bruegel graph. Okay, in this chapter, also I give example how you implement message passing interface. So MPI, in terms of big data computation, is popular and very standardized distributed memory parallel computing. So let's say you have 10 server connected as a cluster in the network. So how you can give tasks to 10 server to compute in parallel and combine again the result of computation into the master node. The common framework for this we call message passing interface. And the good thing in Rust, you can also have the crate for message passing interface. So message passing interface is like master slave, that there is one master giving the task to the slave cluster and collecting the result from the slave when they compute in parallel. Okay, very easy to implement in Rust because it has a good trait called MPI. Okay, you can also play around how to parallelize your code using MPI. So in real work, if you have high performance computing cluster, you can implement MPI to compute in that cluster. And Nextflow supporting high performance computing cluster through slurms. Yeah, you can read documentation in Nextflow how to set up. Okay, but we don't have high performance computing right now, so anything in this book you can run standalone in your laptop. Be familiar with Nextflow so you can make your code portable in the cloud, in the Docker, in the high performance computing. Nextflow is the good tools to reproduce your code anywhere in any infrastructure. So there is a crate in Rust called neatletail crate. This is very hard, memory efficient reading for FASTA and FASTQ file, the raw output of gene sequencing devices. It provides you API to read, yeah? So this is the sample code for neatletail, yeah? So to running this code, make sure that the location of the file path can be found by your code, yeah? Sometimes Rust compile into the different folder where you put your FASTA file in different folders, so sometimes it's not running. But you can easily troubleshoot by asking the AI to find the problem and helping you to locate the location of FASTA file quite well. Okay, just follow the tutorial until you can reproduce the Nextflow project and you can implement basic data structure in RAS for bioinformatics. So we can also see the data or the DNA data sequence as a string, okay, but only contains ATCG nucleotides. But there is a lot of algorithm we know as a string algorithm can be used, yeah, and we can utilize that into biological sequence for manipulating DNA, like searching algorithm in string matching algorithm, yeah. The most famous string matching. What is string matching? If I have a k-mers or some subset of the DNA, I want to match it. How to find the match? Yeah. So the famous string matching algorithm is KMP or Knut Morris Pratt algorithm, KMP. Okay. This algorithm running with worst case time OMN, okay, and knife scan with OMN plus plus N. Okay. So with the AI right now, we can easily implement KMP, yeah, as long as you understand the KMP. So the formal way to explain KMP is using the prefix function, okay? It's also known as a failure function. So for example, if you have pattern P of length M written as P like this, the prefix function P, yeah, is an array which in which P at the position I, yeah, record the length of the longest proper prefix P. Yeah, I think this mathematics explained better than me. So make sure you read the mathematics. Make sure that you understand what it means. If you don't really understand, copy paste and explain more, asking to explain more in your own language in GPT or DeepSeek. Yeah, you can easily copy this text and share to. But before you can copy, you have to be in edit mode in the kernel. Okay, this is the implementation, basic implementation using MPI for KMP algorithm. So you can also use high performance computing like MPI to spread the task of KMP into different clusters. This is the sample code. Okay. Many other algorithms discussed here, like suffix structure, suffix tree algorithm, in string algorithm. Please continue to learn, explore, because if you do your master degree research, everything that you got in this book can help you accelerate your research. So once you read papers, you can easily understand, you have confidence to reproduce the code from papers because you know the basic data structure and algorithm before you're talking about bigger pipeline, more complex process or workflow in bioinformatics. Okay, this section discuss about basic string algorithm. Okay, it consider the DNA sequence as a string and utilizing many well-known string algorithms to manipulate or working with the sequence. So another structure that is important in bioinformatics is graph. If you have bachelor degree in mathematics, you understand that graph defined by G equal V and E, yeah, is a collection of vertex and edges. Yeah, you can see basic graph, and there is some category of graph. We can say it as a Hamiltonian graph and Eulerian path graph. Okay, so you have to know this because this is the basic terminology in graph theory. Yep. Eulerian path is defined if you travel inside the graph, yeah. So Eulerian path is defined as a traversal that use each edge exactly once. Yeah. So if you build the Bruegel graph, it's corresponding to chimeras that have edge exactly once edge. Yeah. So some algorithm utilizing Eulerian path and Hamiltonian path. So you have to understand this term. This is like category, two big category in graph that we utilize its characteristic. Yeah. So genome assembly approach the graph or utilizing the graph in many ways. So once the data loaded into the graph, there is many algorithm that you can run and perform inside the graph, like finding nearest path from one sequence to another sequence, or finding cyclic inside the graph, okay, or find paths on specific long sequence. Because graph already, many people working with graph algorithm before, there's also very important, not only thinking like a string, DNA as a string, but think of DNA as a graph structure, help you to utilize many graph algorithm for genomic. Okay, this section discuss about that and give you some example like how we can build the Bruegel graph from FASTA file, FASTQ, and using little crate to parse the sequence, or using pet graph to represent overlapping chimeras inside the graph. So you can enjoy this library because it's already there in the crate. So you can also try to implement that in this code. Okay, next. Because the biological data set is a very large and big data, searching will be problematic. Imagine a book, textbook. Usually we provide index in the textbook. So if you want to find something, you go to the index and you can locate the pages in the book discussing about what you're looking for. So index and searching in the large biological data set, imagine the data set as a book, big book, is a problematic if you don't have strategy on implementing good indexing, because the search will be quite tough, take it long time. So this section explains the problem, like how to implement common index, like FM index, including the Bureau-Wheeler transform. So many algorithms here discuss about indexing. So as a code sample, you can make a Bloom filter implementation, read the input file, split the reads, okay, in parallel way using Rayon, and sequentially insert this k-means into Bloom filter. Yep. Next, we discuss in the last of this chapter how to perform high performance computing and parallelization. Okay, so as a Amdahl law, parallel speedup can be expected by this equation as equation number 9. Yeah. So this is the basic theory of the parallelization, that if you compute in sequential manner, uh, you can accelerate the computation by doing that in parallel way. You can do it using MPI, or you can use it using multi-core using Rayon. So many types of parallelism. You need to take parallelism course, but in this book, we assume that you don't understand that. So we give the sample code using RAS for you to quickly implement parallelism. So multi-core means that your PC have many CPU core. let's say if you buy new PC and they say is a multi-core, and then you can imagine that there is many CPU in that PC. Yeah. And the computation in that PC is happen in those many CPU. And you can manipulate to make sure that your core utilizing all of the CPU. But if you imagine the CPU is separated server in cluster in network, so we are talking about high performance computing. So for single instruction, multiple data, you can do inside your laptop or inside the SPC cluster. Okay. Nextflow supports SLAM or this is kind of SPC SDK. Yeah? You can read in Nextflow documentation. So implementing basic parallelism, let's say if you want to read large file concurrently in your laptop using multi-core, so you can implement using Rayon. Yeah. So make sure you know how to parallelize the code for reading, because reading a very big file like 10 gigabyte will take time in your laptop. The next problem is how you store that 10 gigabyte into efficient data structure so you can manipulate the data. Okay, so in the last section, 3.6, I just want to show that if you can utilize Nextflow and Rust together, your code will be more scalable. So for example, there is one process you need to fix. You just focus on that process in Rust. And you can also utilizing Python process or C++ process. Okay. This is the tutorial to make example that utilizing Rust and Nextflow together. In bioinformatics, there is so many library already written, and there is no reason for us to rewrite the code. Yeah. So let's say EBIS. This library handle problem that the Brueggen graph have. So this is already MPI ready. Yeah, it's very high performance. Many people use it. Yeah. So EBIS is de novo sequence assembler. So it's developed to handle the influx or of short read by long sequence from Illumina. So is it the assembly by short sequence? Yeah, it's very high performance written in C++. So assuming you install EBIS in your laptop or in your machine, you can call from Nextflow. Yeah. So you can contain the pipeline in Nextflow to call EBIS, to call your Rust code or Python code all together with consistent way. That's why we introduce Nextflow as a part of the pipeline development in bioinformatics. So this is the example how to do EBIS pipeline in Nextflow. Okay. That's it for this chapter. To learn more, you can just copy paste this prompt into ChatGPT and DeepSeek or Claude. I recommend each of you have one at least subscription. You can share with your friend if that is too expensive, like $20 per month. You can join your friend, like 5 friends, and then have GPT or Claude or DeepSeek. Yeah, or you can use the free one. Okay, so I hope this prompt helped you to learn more from this chapter, and you can also learn sample problem in here that maybe your mid-semester or final exam, we will take some problem in the assignment in this chapter or other chapter. Okay, so in each chapter, I give at least 5 problems. Yeah. So this latex is not good. Let me fix. Okay. So how to work with this using AI? So just copy paste, okay? And then you can say, analyze the problem and do research to solve the problem. In GPT, you can use O1 version, which is O1 overview advanced reasoning, mathematical reasoning, or and you can also use deep research to make sure that they looking the solution in the paper, like analyze the problem and do research to solve the problem. Okay, find paper reference and cite the reference into the technical report. Okay, give the code in RAS to be reproduced. Okay. I wish I have this tool when I take my master and PhD. It will go to internet, find papers, and working for you. It takes sometimes like 3 to 5 minutes, but it's like your research assistant working on problem solving, reading papers, finding sample code. Okay, so that's why I recommend you to have at least one subscription that you can share together so you can enjoy these kind of features. It takes time until it's done. Okay, we don't have to wait, but the result is very interesting. You can get the paper, you can get the code, you can get the mathematics, and helping you to be more productive. Okay, that's it for me for today. Uh, Next, I will hand over to teaching assistant Fadin. There is a Nextflow official tutorial on top of the document that I sent in the WhatsApp group. Fadin will show you some basic flow in Nextflow. And before I hand over to Fadin, do you have any question? Any question from you? Any difficulties on RAS and Nextflow from Jonathan, Majere, and others?
Mm, Thank you, sir,
No problem.
and good afternoon. I just want to acknowledge that, yeah, during the first encounter with Rust, it
Mhm.
was a bit difficult, but I'm happy now I can work on it. I'm
Oh,
now able to do it, but the pace is
mhm.
still slow. But now I managed to do the the coding and pushing the work. So I'm glad.
So you can do that now, right?
Yeah, now I can now understand what we are coming from and where we are coming from and where we are going.
Okay, good.
Thank you very much.
So you can also take some tutorial in YouTube if you need. So many Rust tutorial there. Yeah.
Oh,
But you can depend on GPT to write any codes you want. Yeah. As long as you already understand the basic Rust language.
okay,
I
so
su I suggest that you take this book. I think this is the best programming language or Rust programming language even written. This is written by my teaching assistant in Telkom University.
okay,
Okay, this is
okay,
teaching you the basic language,
okay.
but
Thank
you don't have to finish the book. You just need to understand function, how to implement function, and how to compile. So maybe part one is enough for you. Yeah.
okay, okay. So another challenge pack is sometimes I go to the BGVR book,
Uh-huh.
then in edit mode, I try to copy some codes, but
Uh-huh.
I may
And
not be
cannot
able,
copy?
yeah, I may not be
Okay.
able to copy even if I am in editing mode.
Okay, so let's say I will show you here. Let's say I save the chapter 3 and I edit. Okay. So let's say I want to take this code, right?
Yes.
So sometimes the loading, yeah, that one is already loaded. You have to double click somewhere, yeah, and make sure you can navigate here. And then you control command A, copy, yep, and then paste. Okay. So
Okay.
that problem happened because you have to double click somewhere until it's like refreshed, and then you can copy here. Yeah.
Okay. Okay. I see.
Okay, and then save again. So you
Okay.
cannot copy if you're not in the edit mode.
Okay.
You have to double click somewhere near the code until it like refresh, and then you copy the code. Yeah.
Okay. Thank you. Noted. Thank
And
you.
me by proofreading, sometimes LaTeX is not well in GPT, and so it uh, still some typo. Yeah. If you find typo, go to edit mode and then uh, fix the typo. Okay. Sometimes it print the latex 2 times.
Okay.
Okay. Help
Okay.
the error this one. Yeah. This M should be latex. Means that I have to put okay. Latex in curve note is dollar. You have to type dollar and close with dollar.
Okay.
Okay. Okay, that's it from me for today, and please continue with Fadin to rush to Nextflow official tutorial. Please, Fadin, takes your time until the tutorial is delivered.
Okay, thank you so much, Mr. Risman, for delivering the chapter 3 of our books. I will continue the lectures about Nextflow introduction. Okay, so I will start. For Nextflow, we can run Nextflow in our laptop if you already install the Nextflow. So if you go to nextflow.io, you will see this type of code step set for Nextflow, which have some parts called process, and in each process have input, output, and also have workflow and also have channels. And just to make sure you have download this prerequisites, Java, and also install the Nextflow in here. And if you haven't, don't worry, you can also use GitHub code space from Nextflow training tutorials. So in Nextflow, they have dedicated training resources that you can go to training.nextflow.io. In here, they have multiple resources for Nextflow, right from the beginner, Hello Nextflow, and for newcomers, and also in-depth Nextflow training, the fundamental one, and also the advanced training. So you can go in this page. I will send it in the chat box to see the training Nextflow. And you can click the open in GitHub code space. Then the Nextflow GitHub code space will prepare for you if you already have a GitHub account. So usually they've been used Gitpod, but until February 2025, however, the Gitpod decided to retire the free functionality and they switched to the GitHub code space to host the cloud tutorials in the GitHub code space. So if you click on this, then you will have the GitHub code space made for you. Then in the file explorer, it already equipped with each Nextflow training materials. So in here, the Hello Nextflow, NF Training Advanced, NF for Science, and others is already equipped for you to do the training tutorials. And you can also write in your IDE browser, example like VS Code or Cursor, if you already installed the Nextflow also. So if you already click on the GitHub code space and the GitHub code space will open up for you, we can you can click Control Shift P and search for simple browser so we can have a trending Nextflow right on our right screen and you can adjust it to the right of your screen. So you can learn by doing with open up the training Nextflow tutorials. So we will continue to launch the Hello Nextflow training in here. Okay, so for the Hello Nextflow, there are several Nextflow training file. This one is Hello Channels, Hello Config that we can try. And also the tips in here. For whatever reason, you have to make sure that you are in correct directory and you can comment in the terminals to change directory to the directory that you want to try the tutorials. Okay, so for part 1 in Hello World, in this training course, we can start to run Hello World directly. So you can, in your terminal in here, I have to change the directory first to the Hello Nextflow. And we are already in the correct directory. And we can copy this code and make the terminal say hello. And the output will have the hello world in the terminal and it is going succeeded. Now we can move on to the second part, how we can write the text output to a file. In order to do that, we just simply running the previous command and we specify that we want to save it in a text file named output.txt. And in here, you can see that there is some generated file output.txt that store the output from echo hello world in here. And we can also show the file contents in here using the command cat output.txt and it will show what is the output file inside the output.txt. Okay, so in Nextflow, we are going to open the hello world.nf. So in Nextflow, the script will have a format file.nf. And in here, they use some part of process and also workflow. So what is process and workflow? This is the basic concepts of Nextflow, and it is the difference between others DAD and others workflow management for bioinformatics. So in Nextflow, channel and process are the core ideas. So imagine a channel is like a pipeline or queue that sends data items here labels as data Z, data Y, and data X into a process, then the process run a task for each item that arises. So in here, the Nextflow will process, for example, task 1 handles data X, task 2 handle data Y, and task 3 handle data Z. And because each data items have its own tasks, all those tasks can run in parallel. So if you have multiple CPU cores or a cluster, Nextflow automatically can orchestrate this task. And each process typically outputs something like a result file and also some metrics. Then you can feed those results into another channel to keep the pipeline going. So putting it all together, you can continue to divide another channel process and the workflows. So let's go back to the GitHub code space. And in here, we can try to running the hello world.mf. In here, there are one process called say hello, which will have an output with the file name output.txt, and the script is will print out the hello world. And in the end, we have to create a workflow that running a process, which is the say hello process. So in order to do that, the code is already defined. We can run it by Nextflow run hello world.nf. So we can copy in here and paste in here. And Nextflow is running the hello world.nf. So in terminals, you can see in here Nextflow version that running the script and also the executor. And this in Nextflow, they will have a log for each process. So in here, the process, which the process name is say hello 100% working, and it's already executed perfectly. So if you have multiple process, then the process will continue and the Nextflow will show you is it working or have some errors. That's it for the first Nextflow workflow. So moving on, in here, you can also check the console log line for the say hello process. For example, in here, we can see the output.txt. You can see the log, all of the run in here in the work directory. Okay, so moving on, we will continue to the part 2, which is hello channels. And in here, we are opening the hello channels.nf. So in here, there are a workflow with say hello params.greeting. So the pipeline parameters we define in here, params.greeting is holamundo. And let's see if we run Nextflow run hello channels.nf. Okay, then the process is executed perfectly. So here you can try and exploring in the GitHub code space. And also you can do that in your local in VS Code or Cursor. You can just create a folder in your GVR and create a Nextflow, and you can right click on the mouse and new file, which is, for example, hello channels.nf. And you can copy the code from hello channels to check whether it's working or not inside your own local. And in here, we can copy from this and save. And in here, we can type in your terminal, Nextflow run, hello, channels.nf. And it's working. So no matter what the IDE you are running the Nextflow, whether it's on GitHub Codespace in cloud or it's in the local, just make sure that you already install Nextflow correctly and it can run the Nextflow. So if you have encountered some troubles, maybe you have not set up the Nextflow installation properly. So make sure that you have corrected Java version and also the Nextflow version that can run inside your laptop. So we will continue in here. You can try in each of Hello Nextflow, but I will show you how to run a fundamental about the bioinformatics one. To do that, I have to open the simple RNA-seq workflow. So in here, we can try to run this. There are, in NF training, there are several scripts that is actually built built from script 1 until script 7 that running completely for a single simple RNA-seq workflow. So in here is actually there are some parts, but for now we can check in here. So there are already a .fa file in here, transcriptome file, and also the read.fq. This is the FASTQ file, and we can run it by this. We have to change the directory first to the NF training. Yes, and then to ensure we are in the right directory, we can type ls and we are in the right directory, and we can type nextflow run script 1.nf. Okay, then for the script one, it will print out the rich params that are already defined by us in here. We define that we are going to read the projector in here. Um, um, got got file from the FASTQ. There are got1 and got2 FASTQ file, and in it's it can run perfectly. And in here, moving on to the script 2, it is more complex in here that have one process called index. This process will have an input of transcriptomic and output is Salmon. So Salmon is actually the tools that we can use in here. So it's a tool for quantifying molecules known as a transcripts through a type of data called RNA-seq data. So this is actually a GitHub, the open source tools that we can use inside the Nextflow. So in order to do that, we can check it out for running the script number 2. To do that, we can Nextflow run script 2 with Docker. So because the Salmon is from the third party, not inside the Nextflow, we are in the nextflow.config already define the Docker run option. So in here, we can run it Nextflow run script2.mf with Docker. If you not specify with Docker, then the code will run with errors because the tool's not already not inside the Nextflow. So in here, the process will execute the index process, and we will wait. Okay, while we're waiting, you can check on more about the Nextflow training in here. This is complete Nextflow training, and it's beginner friendly. So if you wonder how we can do environment setup, like in the GitHub code space, like if you have not a GitHub account, you can create this and continue the tutorials in here. And also for local installation, you have to have these requirements for Java, Docker, and others, and also download the Nextflow version. And you can try to complete this for Hello World channels, workflows, modules, container config. So this is the basic fundamentals core of the Nextflow. And for genomics, if you're already familiar with how to set up the Nextflow, you can move on to the section of the Nextflow for genomics and also for the fundamental trainings. I recommend you to complete the simple ArenaSec workflow and run and see what are the results in each. Actually, you can run also from a project from GitHub in here. They store the RNA-seq and F in a GitHub, and you can launch the pipeline execution like in here. We can Nextflow run Nextflow IO RNA-seq and F with Docker. Let's try. Nextflow IO. Nextflow IO is in. So it will pulling up the RNA-seq and F from Nextflow IO GitHub. So in here, this is the complete pipeline of a simple RNA sequencing. And in here, there are 4 processes, which are the indexing, the fast QC, and quantification, also the multi-QC. So as you can see in here, the process is executing perfectly. And you can also check, where is it? In F9. F9? Okay. Or A5. A5 in here. Multi-QC. And we can see the report in here. Because it's HTML file, we can see the report of multi-QC. It will reporting the report of the quality checking of our FASTQ file. And here in here we have 2 files, and each in here the someone is reporting a tool for quantifying the expression of transcript, and this FastQC checking the quality control for high throughput sequencing data, and so on. So you can utilize it, and you can also check in their GitHub because their repositories is complete enough in Nextflow IO. And they have also the community, the pipeline, we call it NF Core. In here, there are hundreds of pipelines that is available to run, like SAREC, rare disease, Pangenome. It is a lot of pipelines that community is contributing to NF Core to upload the pipelines. So if you have some cases, some research that is bioinformatics area, you can try to check in NF Core, is there any pipelines that suitable for your research, and you can utilize the NF Core. So that's it for the Nextflow introduction and NF Core. If there are any question, I will glad to help to answer. Any questions so far? Okay, so if there are no questions, maybe I will stop in here, and I will